Hi everyone. It's a really great pleasure being here. I hope you enjoyed so far, as I do, for sure. This is the title of my talk. I want to somehow give you an idea where we all use F# in our daily business at University of Kaiserslautern. So computational systems biology, this is the title of our group. We are interested in how cells change their molecular structure if you give them an input like environmental change. And we are particularly interested in how plants behave, because they cannot run away. Yeah? If it becomes hot, yeah, they really have to deal with this. And dealing with this means like they really completely have to change their molecular structure, the complete network, to have a response. And this is what we tried to figure out. And Benedict gave you already a small introduction in how we're going to do this. First, I want to introduce you to the team actually who's doing all the work. They are all forced to do F# willingly, which is great. And they are all here, so if you want to talk to them, they are there. In the middle of the Palatine Forest, there's an outpost of F#ies. That's us, that's our building. There we are located. So whenever you're around and feel like talking about F#, visiting us, there we are in the middle of the forest. Nobody can find us if you do not know us. Anyhow, what do we have tasked in academia? So they are divided in, of course, teaching and research, and our research is again subdivided in data science, and in this audience I should not call it software development, I call it method development. Okay, this is our task, and this is how it is divided. And I want to, because it's the data science conference, I want to go into this a little bit deeper. And we most of the time have this classical cycle of data science. We have to process the data, we have to analyze the processed data, we have to somehow visualize, and then generating models and hypotheses out of it, and then it starts again, the cycle. And in research, especially in bioinformatics, these are the languages, and I try to fit the symbol to the size the languages are used, and F# is not on this map because of sizing at the moment, but this will rapidly change in the future, I'm 100% sure of it. But you see here another problem. It is quite a mixed bag. So the data processing there are for sure is just not fast enough because Matthew did not work on it, obviously. So there's different languages involved, and we are using F# to homogenize this zoo. And we do this out of different reasons. But of course, the biggest question from students I always get is, why F# when all my friends are doing Python, right? And of course, I had to think about this a lot because I'm strong opinionated about F#. The person with beautiful hairs want to discuss about this with me later, but we will do this. So why do I'm keen on F# is because this is the curriculum in undergraduate biology. All the gray boxes are my colleagues, and the purple box, this is me. So this is the amount of time I have to teach them concept, language, and also, of course, bioinformatics, right? So I know it's good to switch languages, but if you have to fit everything in there, and nowadays they really need to go through this, so this is great. But this is why I decide just to take one language which fits best to all tasks. And of course this, right? Okay, so I really looked at what we are really doing in bioinformatic data science, and if you look at it, we always start with a measurement of a certain kind. You see a mass spectrum there, I will explain in a minute what this is, or some sequencing data, then we process it, making like value tables out of it or data matrices. Then we do statistical analysis, and then we integrate this and build hypotheses. And of course, you immediately see that this is just information transformation by function, information transformation by function. So map reduced, basically, right? And if you once understood this and once grasped the concept which Thomas elaborated on, that this functional composition, every one of you knows even better than I do, is that you really can break very difficult problems into small functions, smaller functions, and then you can basically solve it all. It's like the skeleton key, right? And this is how I also refer. I like this pipeline idea. It's super obvious, of course, but this is how I do it in my lectures. And I did, I don't know, 25 video lectures, like teaching F# in German for beginners, like really non-experience of computation or programming whatsoever. They are out and are used. Then what we do is we try to write a lot of blog posts in FS Lab that we start to build up a resource for students, because we learned, I mean, one of the biggest advantage of R is that you can copy paste your solution and change a string, right? We are gonna get there. Exactly. The next thing is what we do is we offer student projects, which then sometimes even end up in publications, which is really nice. So, and the other thing is what we are heavily using is notebooks to teach bioinformatic processes. So you see here on the left, there's a notebook where we use ODE, so ordinary differential equation modeling, to model how a cell, how a transcript level is behaving when you introduce sugar into the cell, and what is happening. And this is just done by ODE and solved by Runge-Kutner or Euler or something. And then the people can really play around with this. Today I want to go with you, because I cannot teach you anything about F#, you are the experts here. I just want to go to through one of the workbooks and just show you that you get an impression. So first you need to know, Benedict already explained a little bit, that proteins are like the key players in living systems, right? They do basically everything. They are involved in every process. They are like the operators. And if you think functional, then you would say they are the function of living systems, right? They transform. Okay, what you also need to know that these functions are encoded in something what is called primary structure, which is a sequence of smaller molecules called amino acids. The problem is that when we start with data, then they are most of the time untyped. What you see here is AGTACA. This is a sequence. This is how we get it from a reader. I will show you in a minute how this works. But then this could be either DNA or protein, depending on how you interpret it, and this makes it very, very different. So the result is very different. And therefore, of course, the first thing what we want to have is a function, which the function takes this ambiguous code and transfers it to a type safety, what we love in F#, right? And this bioarray of amino acid string then tells it, okay, please interpret the string as an array of amino acid, and this is what you get, right? This is somehow the starting point, and this kind of functions you find in a library which we started. It's BioF#. It actually was my first attempt in F#, and these guys over there, David, Kevin, and Johnny, they continue developing this. So there you find all these functions to really model biological processes and work with biological data. And I want to introduce today to you something which is called proteomics workflow. This is the way how we measure the quantity and the identity of proteins. And therefore we use a device which is called mass spectrometer. And this is, you can imagine this as the world's best scale ever. nanoscale. But the only thing what this can do is it can tell you how much a thing weighs. Okay, but what we want to do is we want to know the identity and the quantity. The classical thing is that there's a sample with like thousands of 50,000 of proteins in. They are extracted in the lab. Then you have a mixture of proteins. Then you give it another protein on top, which then chops the protein in pieces. The pieces are called peptides, and they are injected into this mass spectrometer, which is like a vacuum scale, also a scale in vacuum, and it gives you these spectra on the right side, which gives you the mass of the whole thing, and then it collides with other ions and even damage it. Yeah? And then it gives you a fragmentation spectrum. And these are the two information you have, like the whole thing, so the whole peptide, and the fragmented peptide. Yeah? So this is what you get. So a scale which weights and then damage everything and weights the damage. Okay? This is what a mass spectrometer is in a nutshell. So I want to model this first process, which is the digestion, so chopping the protein into peptides. This is done by another protein, and as I said, we model proteins as a function. So we have this digest function. What this digest function need to know is what's the name of the protease, because this is the pattern how the protein, what you also give into the function, is chunked into pieces, and what you get out of it is peptides. So on the top you see the symbols of it, and on the bottom you see the F# code if you use BioF#, right? So you have this digestion.bioarray digest with trypsin and give this thing an identifier, and what you then get is you get a list or an array of smaller sequences called peptides. Okay, great. Then, so we modeled this part here. Now it goes into the machine, right? And then we measure masses. So what we know is the organism of which we get the sample from. So we have, because we know the genome of it, so we know the sequence of the potential 50,000 proteins which we might get. Okay? So we can take this sequence, convert it into potential protein sequences, digest it with the function I just showed you. We just filter for outliers because our detector cannot go over 300 mass units, 3,000 mass units, but forget about this. And we make a histogram with plotly.net, and what you see here is now all the masses, a histogram of the masses which you can expect because you know the organism. Okay? Cool. Good. So we have all the potential masses we can get out here and can compare this list now with the masses we really get from the machine. So we have like a first filtering step. So the next step I told you is that this scale does not only weight our stuff, it also breaks it into pieces by ion collision, like random pieces. And the random pieces are called B and Y ions, depending on the fragment identity. So this is the next step we need to model, right? So, of course, there are functions available which simply chopping this into pieces, calculating the mass of the hypothetical ions and producing a spectrum, okay? And we expect from the machine that it gives us the same amount of Y and B ions. This is just experimental evidence that we believe. So we can produce something like this. So we produce a hypothetical spectra just by completely modeling this process in F#. And so what we do then is two other steps. So we take the real measured spectrum, we pre-process it, so meaning cleaning it from noise. We know the mass of the whole thing, right? So we select out of all potential peptides, the one with the right mass, to overall mass, right? And produce this for every potential peptide, we produce, as I just showed you, a hypothetical spectra. And then what we do is we match the pre-processed spectrum with the hypothetical spectrum. Okay? Everything in F#. And this is how the spectrum processing looks like. So we get some file, which if you plot the file, looks like this. So this is a real-world spectrum from the measurement device. We know the scan limits, so we know from where to where the machine can give us the correct weights. And what we then do is we pre-process it, and there are various methods of pre-processing this kind of spectra. This one I show you is that we put it into chunks and normalize every chunk with the maximum peak to 1. This is also empirical, that the matching algorithm I show you in a second matches this best. Okay, good. How do we match? We use autocorrelation. It's an algorithm which is called Sequest, which just does the correlation divided by the autocorrelation of the theoretical spectrum with the real spectrum. So what is autocorrelation again? So we're not only measuring, we're not only like taking the sum of both peaks at the same place or same mass, but we also move it across the spectrum and get a correlation value on every shift, right? And of course, there where it fits best, we have the highest peak. And if everything fits random, you would expect something like this, right? Like a random line. So by dividing now this upper part through the real match with 0 shift, we get a correlation value tested against random match. Okay. This is also done here in this line of code. So what do we have here? So we have the peptide masses. We filter the sequences according to the mass because our precursor mass is between these two numbers. So we get only the possible values according to the full length mass. Then we simulate the ion spectrum with the function we just wrote. So all the B and the Y ions, right? And then we do this autocorrelation by sequence like scoring a single spectrum, the theoretical spectrum, against the processed intensities. So we get out this array here, and what we get is all the possible peptides which could match and their sequest score. And what you see here, this one is 11.4 and the next one is 4.8. So because of this big difference, we can be quite confident that we identified the right peptide, right? So I think quite, I don't know, heavy process, completely modeled in F#. And we really use this in production. So this is what the students do, like writing a small piece. But we have this also in a more performant variant where we really process our data with. What you might have seen is that this is our theoretical spectrum, and you would have thought that this is maybe correct because you have like the same peptides from a protein should have equal height because they should be in equal amounts. But this is the problem or the difference between theory and practice, right? In practice, it looks like this. Even though they are in equal amount there, they have different heights. And so far, what I showed you, we did not take this into account, so our very naive modeling made all similar, so all intensity heights the same. I don't go into too much detail, but of course we develop also an algorithm for this. So we use, it's called DPOP, which is a deep learning algorithm. We did this 2018, okay, so a little bit time ago. But what we did is we converted this sequence into numerical vectors by projecting it into the physical-chemical properties. So this is representing the physical-chemical properties of this particular peptide. It's like a physical-chemical footprint of the sequence. And then we could learn the observability when a peptide has this kind of physical-chemical properties. And we use a neural network, a very straightforward neural network, to learn the relationship between the physical-chemical properties and the peptide observability. And this is a little bit what I mean when I said software development at the beginning. What is then very nice that we can stay in F# and offer our people or our colleagues from the lab a website where they can then predict it. Of course, we appreciate SafeStack a lot for this because we can just go on and design a small page, and behind in the back end there is the algorithm running, and the people can really use it, which is very nice. And we do not have to switch language, which is an important point for us. Okay, with a little bit time left, I really want to relate what Evelina was talking about, because I completely was nodding all the time, I guess, in your talk. What we did not talk about yet is that where did the samples come from, right? From there, everything is very easy to model. Everything is easy in F#, but really knowing what sample relates to which file and spectrum later and what the biologists did giving us the samples in hand is really a bigger problem when one can think, but Evelina already elaborated on this. So, but we took a little bit a different approach in a big consortium we are in. We do not simulate the data, which is great actually, but we want to somehow give the experimentalists tools at hand that we can communicate better what happened with the sample and how this was produced. And therefore, we say, okay, everything is a process. You know, the experimentalist has some material, then there's a process applied, one or multiple processes applied, then samples come out, then a measurement process is applied, and we have a measurement, and we have also some administrative metadata, which means like who did the thing, right? Of course, you cannot say, please give me a acyclic graph which represent your experiment. So we collapse this into a table, and we always say, okay, tell me the source name and the sink name, and put columns in between, which describes the process from source to sink. And the important bit here is that you should not use free text, you should use an ontology. So you should really use the word instrument model, write it like this, and if you measured it with the device, the mass spectrometry I just showed you, it's a triple TOF 4600, you have to write it like this, right? This is of course challenging, and what we did there is, this is mainly done by Kevin Frey, but also Lucas and Oliver from my group. This is a tool which we call Swade, and again here, thanks to SafeStack and of course Fable, we could program an Excel plugin which somehow searches in an ontology database for like fitting vocabulary, which you then can automatically transfer into this table, which make like typing errors and so on, and thinking of what should I type in this table much easier for our experimental colleagues. Yeah? And there are people who love Excel, which use this one, but there are also some people which are strongly opposed to Excel. Therefore, we have also a web-based free version of the same thing at the moment in the alpha state, but it will come out as fully usable. And this is what you then produce, and I just give you a screenshot here. So if you type instro, you see that it suggests you instrument model. this ontology term has an ID and is therefore absolutely identifiable, and you can really use it. Yeah? We also, because these vocabularies called ontologies have always the problem that it's somehow speech and they are never complete. Yeah? And this is a big problem. And the way we solve this is a very low-tech idea, is that we say, okay, before you write it in manually, please make a pull request on a simple file on GitHub. Write the term you are missing into this ontology, like a term collection, and immediately when you go through with it, we use continuous integration, this parsing this into the database, and it's immediately available for you and also your coworkers in the Swade tool I just showed you. And by this, we really could crowdsource the controlled vocabulary development, which is like a tremendous pain, and if you hire people only doing this, they don't know what the other people are talking about. So this is actually something which really is solving a problem for us, which is a much bigger problem than I thought at the very beginning. My group again, doing all the work, super cool. I'm done. Thank you for your attention. I hope you enjoyed it.